Hi, everyone. Thank you all so much for coming today. So over the summer, I conducted a project in which I explored the impact of the addition of a short-term gluten-free diet on the microbiome of people with Crohn's disease. So first and foremost, I would like to thank Papa Jack. And for those who don't know, Papa Jack was a biology teacher that inspired his students inside and outside of the classroom to pursue scientific exploration. And it's through his funding for this fellowship that doing this kind of research is possible. So now moving into to provide some background on my presentation. Crohn's disease is a chronic inflammatory disease that can affect any portion of the digestive tract, including the mouth, esophagus, and intestines, and the ileum as well. And it is also an autoimmune disorder, so the body will attack its own tissues and cells. So in this picture here, we can see in the person with the normal intestine that there's no yellow fat that wraps around, whereas somebody in Crohn's disease, that yellow fat wraps around the intestine. And this yellow fat can often lead to symptoms like diarrhea, abdominal pain, rectal bleeding, fever, and bloating as well. And many of these symptoms impact people in not only their personal lives, but their professional lives as well, as they're not able to obtain a job because they need vicinity to a bathroom or toilet. And these can plague them for the rest of their life. So in order to help alleviate symptoms, individuals with Crohn's disease often need surgical intervention at least once in their lives. So during surgeries, the surgeons will remove the damaged part of the digestive tract or the intestine and then piece together the healthy parts. However, I want to make clear that surgery is never a cure for Crohn's disease, and that oftentimes pre and post surgery, a medicine called Remicade serves to reduce inflammation in people with Crohn's. However, when going through the process of Remicade, it can be extremely painful as you have to endure sharp needles piercing your arms, and the long terms of Remicade can be bad as well, as there can be an associated loss of response or malignancy, as well as feverish and flu-like attempts. So in order to alleviate the long-term effects of surgery and Remicade, you can adapt special diets, such as a gluten-free diet, which is a commonly adapted special diet to help individuals with Crohn's disease or gastrointestinal complaints. So a gluten-free diet eliminates the consumption of anything made from wheat, and it's been used in people without any underlying conditions, and it's been found to benefit them and their health long-term as well. So when changing the diet of somebody, and especially somebody with irritable bowel disease or Crohn's disease, you're bound to see changes in the gut microbiome and fecal microbiota. The microbiome is a collection of microorganisms such as the bacteria, viruses, and fungi that live on or inside the body of a specific environment. And oftentimes the microbiome tells you a lot about the health of an individual. So the gut microbiome reflects health, and oftentimes a gut microbiome tends to imply health when there's increased diversity. And there are certain families of bacteria within the microbiome that might be more healthy or unhealthy for the body. So one family of bacteria is the Lactospiraceae family, and the Lactospiraceae family utilizes hydrolysis of sugars and starch to produce butyrate. And butyrate acts as an anti-inflammatory agent. So over here, we can see the pictures of the bacteria and what it produces on the right. So butyrate is known as a short chain fatty acid, and when it's produced, it breaks down fibers in the large intestine or colon, and it helps to control inflammation. In previous studies, the Cleveland Clinic and Mayo Clinic have sponsored studies in which they provide butyrate supplements to people with IBD or ulcerative colitis or Crohn's disease, and they've noticed a decline in symptoms while on the supplement as well. In addition to Lactospiraceae, Veillonella is considered to be a pro-inflammatory of bacteria, and an increase in abundance was consistently reported in IBD patients. So from this preliminary research I did, I identified two research questions and a hypothesis. My first one was, how does a short-term gluten-free diet impact the symptomatic expression like bloating and abdominal discomfort in people with Crohn's? And my second one was, how does the short-term gluten-free diet affect the gut microbiome of normal individuals and in individuals suffering from Crohn's? With sub-questions, how does the diversity change? And are there any specific families of bacteria that change as well? I hypothesized that a short-term gluten-free diet will help to reduce symptomatic expression while making beneficial changes to families of bacteria for individuals suffering with Crohn's disease. So after this, we designed a methodology starting with recruitment and pre-approval. So we recruited a normal individual and an individual with Crohn's disease to participate in the study. IRB approval was obtained, participant consent was received, and then I communicated with the UH Mauna Kea lab and Iolani Wet Lab to schedule times for lab work. So here we detailed the study methodology. So we had the first 35 days as a baseline. So on days 0 and 35, we took a stool sample and we kept a survey result going throughout the entire course of the 35 days to track symptomatic expression and food intake. And then we continued on to the experimental period, which is 35 days of a gluten-free diet. And that's also, we stool sampled on day 70 and we continued with the form every single day as well. And then we went into the washout period on the last 35 days where participants started to increase gluten consumption like they did in the first 35 days to see how gluten-free really impacts it. And then also on day 105, we stool sampled and we kept a survey. I used a survey every day, the same survey to track symptomatic expression. So here's the survey that I used. It was quite simple. I tracked basically just lunch, dinner, food intake, what the ingredients contained, and the symptoms, severity of symptoms, and stress level as well. So then going on to the microbiome, we started with stool collection. So starting with stool collection, I used a piece of saran wrap to prevent contamination of the stools. And then I beat the stool samples in order to break down cells for DNA extraction and microbiome analysis. And the last process I used was filtering, which is a series of washes and vortexing in order to further enhance the cells for DNA extraction. After we did this, I was able to go on to the DNA extraction process. So after elucidating the DNA, I used a Maestre-Nagel 16S DNA stool extraction kit and was able to extract DNA from stool samples. After that, I used a PCR reaction, which stands for a polymerase chain reaction. And this is a process that amplifies certain sequences of DNA for replication using a series of denaturing, annealing, and elongation. And the last step in the process was DNA sequencing. So I used an Oxford nanopore sequencer in order to get a read on certain classes of bacteria and how the abundance of these bacteria changed over time. So moving on to data and discussion, the first thing that we can discuss is symptomatic expression. So using a McNamara statistical analysis test, I analyzed days experienced symptoms before and after a gluten-free diet in the patient with Crohn's disease. So the symptoms I tracked were things like bloating and abdominal discomfort. From the daily survey results, we can see on the left from before and after that the patient with Crohn's disease experienced less symptoms after completing the gluten-free diet. As before the gluten-free diet, we can see that they have a symptomatic expression day of about 10 plus, around 12 days. And then after the gluten-free diet, it drops to about 2 days. So after doing this, we achieve a p-value of 0.043, which indicates statistical significance in this part of the study. Next, moving on to the microbiome, we can also see that the diversity of the microbiome declined during the gluten-free diet, a period of time in which the experimental group experienced less symptoms. So we see here, as outlined by the 2 blue dots, which is the experimental group or the patient with Crohn's disease, between time points 2 and time points 3, when they were on a gluten-free diet, we see the diversity of the microbiome change or drop dramatically. And this is surprising because, as I mentioned before, a more diverse microbiome normally indicates health. But when the patient was complaining of less bloating and experiencing less bloating and abdominal pain, they experience a less diverse microbiome. And now the last thing is the principal coordinate analysis. And this reflects a shift in the composition of the microbiome. So how bacteria classes change and which ones are introduced or eliminated from the microbiome. So at the first point, we can see in the experimental group, as illustrated by the triangles, it's at negative 0.2. And then after the first baseline, it's going to go up. And after gluten-free, it's going to go above 0.2. So we're noticing a significant shift. Now, when we return to washout and the experimental group starts to eat gluten again, we can see that the composition of the gut microbiome is going to drop back down to what it was originally. So from this, I determined that gluten played a major part in shaping the gut microbiome of an individual with Crohn's, as the removal of it results in a shift and the reintroduction of it resets it to its original state. So then we can also see that there was an abundance in the bacteria family Lacnospiraceae while on a gluten-free diet. So as previously mentioned, Lacnospiraceae was a anti-inflammatory family of bacteria that produce butyrate, which is an anti-inflammatory agent. And you can see here in the control and in the experimental between time points 2 and 3, that you can see that for the control, the abundance will go from 51 to 69. And for the experimental, it's going to jump from 39 to 51. experiencing a significant increase in this family while on a gluten-free diet. And the graph on the right also aids in this, as you can see an increase in the abundance of the family while on a gluten-free diet. In addition to Lactobacillus, we also saw a decrease in the bacteria family Valenaceae while on a gluten-free diet. So as previously mentioned, Valenaceae is a pro-inflammatory family of bacteria. And we can see here between time points 2 and 3, the experimental group, the abundance of that bacteria dropped from 25.5 to 0.7, which shows a significant decrease in the abundance of this family. And this is aided by the graph on the right, as you can see the decrease as well between time points 2 and 3 for the experimental group in this instance of bacteria. So after analyzing all this data, I formulated a lot of major conclusions. The first one being that a gluten-free diet helps with reduction in symptoms like bloating and abdominal pain. The diversity of the microbiome did not increase during a gluten-free diet, but rather decreased. A gluten-free diet increases the abundance of the bacteria family Lactobacillus, which in turn helps with butyrate production, an anti-inflammatory substance. A gluten-free diet decreases the abundance of the bacteria family Valenaceae, which is a pro-inflammatory family of bacteria. So after analysis, I drew a conclusion that the abundance of certain families of bacteria matter more than the diversity of the microbiome when talking about the health of the patient. And I drew this conclusion because the identity of the microbiome, like we saw a increase in Lactobacillus and a decrease in Valenaceae, that impacted the patient's health as they experienced less symptoms than the diversity did, because that diversity decreased while certain families of bacteria, the prevalence changed, and that impacted the symptoms more than the diversity in regards to Crohn's disease. So in regards to future research, the first thing I would like to explore is DNA methylation. DNA methylation is a tool that helps to regulate gene expression and analyze inflammatory response. So you can kind of see if certain genes, if they're pro-inflammatory or anti-inflammatory, are turned off or on while on a certain gluten-free, on a diet, and see how the presence of these genes impact the patient's health. You can also see cytokine levels, and cytokines are proteins that are produced from the genes, and levels of cytokines are often regarded in regards to inflammatory mediation and triggers in inflammatory disease as well. And just to draw an important distinction, DNA methylation and cytokines are easily quantified and evaluated from blood. So the microbiome that I did over the summer was done from stool samples, but this will be done from blood. So during the 35 days in the previous workflow I mentioned, we also drew blood as well to aid in this future research. So some takeaways from the Kay Fellowships. My first one would be that doing research is about learning new things. So for me, it was about learning like coding and using different languages and stuff like that. And so it was like not being able to, you have to be willing to try new things and not being able to confine yourself to a box. And then my second one was persevering and adapting to various roadblocks. So things are very common to change at the drop of a hat. So you have to be willing to adapt and persevere. For future K fellows, my advice would be to stay organized. So write everything down in your lab notebook and organize your articles and citations, as it makes it a lot easier to put together these slides or write the paper in the long term. My second one is be persistent. Like I mentioned before, it's really important with whatever changes might come your way or whatever results you may or may not see. My third one would be to have fun. You're going to be working on this project for a long time. So it's important to really enjoy the topic you choose and be passionate about it, because otherwise it's not fun doing it. And then quantify data as much as possible, because it's a lot easier for analysis. And then the last one would be to detach yourself from the results and enjoy the process. So you're obviously going to get a result, and sometimes it's not going to support the hypothesis, but you have to be okay with that and kind of just enjoy the process, because you're always going to prove something in what you do. And my last one was acknowledgement. So thank you to Papa Jack, the Mauna Kea Lab, Iolani School, and the Iolani Wet Lab for providing me with the facilities and resources to pursue this kind of research. To my participants, thank you for taking part in my study and being willing to adapt to the various diets and the procedures as well. To my family, thank you to my mom, dad, and sister for putting up with my request of eating gluten-free for 1 month and being a pillar of support for this entire process. Even though you guys didn't let me store poop in the freezer, this project would not have been possible without all your love and support. To Ms. Kobayashi, Thank you for always encouraging me and supporting me throughout this process. To Dr. Jane, who couldn't make it today, thank you for all your help and support in regards to creating the form and tracking symptomatic expression. To Dr. Chan, thank you for encouraging me to keep going and reading the literature to find a gap, even when I didn't want to. And to Dr. Perez, who couldn't make it to the presentation today, when from reading my research proposal, he immediately lended all support and guidance. He came in on holidays to draw blood and work with me in the lab. So much of this project couldn't have been possible without him. And last but not least, my main mentor, Mr. Hill, thank you so much for everything. You all won't know the half of it, but from Mr. Hill has helped me guide me through every single step of this project, from the research proposal to these slides, and I'm incredibly grateful for all the support at every point in this journey. Also, thank you for catching me when I accidentally transferred DNA into the wrong tube of liquid. I know I never lived that one down, but who knows what data I would have been presenting if it weren't for that. So that's it for today. Here are my image citations, and do you have any questions? Thank you all for coming.